And the last species of Clostridium we need to discuss is Clostridium difficile, or we call it C. difficile. It is classic Clostridium with oval spores located subterminally. That bacteria, as any other Clostridium, is widely distributed in soil and water with the help of spores, and actually it belongs to normal flora of intestine in humans, in animals, in both. And we have two types of Clostridium difficile, toxigenic and non-toxigenic. Usually, strains belong to normal flora of human, are non-toxigenic, and it is kind of fine to have that bacteria in intestine because it's not dangerous for our brain. But toxigenic strains can produce two important toxins, toxin A and toxin B. First one is an enterotoxin. It can affect intestine and lead to serious complications in the type of pseudomembranous colitis or antibiotic-associated diarrhea. And cytotoxin as well can damage cells, directly leading to serious damages and worsening duration of mentioned complications. As well, some protein was identified which can inhibit peristalsis of intestine and lead to accumulation of toxins in that and accumulation of bacteria in intestine of affected person. And usually that bacteria is the most important in clinical practice because you will deal with it more frequently than with any other species. Bacteria is observed in people with prolonged antimicrobial therapy and impaired immunity. Usually it is a patient hospitalized in some intensive care unit department or any intensive care ward in usual department when common status of subject is bad and antibiotics are prescribed in some long duration, massive dose, or in combinations. By symptoms, diseases vary from simple watery diarrhea, which can be observed in some 3 episodes per day or more, and it can last for few or more days with common symptoms of infection such as fever, loss of appetite, different problems with intestinal peristalsis, bloat in abdomen, tenderness and pain, abdominal pain, and other symptoms. And usually, specific treatment is not required because bacteria can be easily suppressed by normal flora, and the main task for doctor is restore normal flora. Again, the main reason is incorrect or administration of antibiotics or some surgical invasions on abdominal cavity and intestine. And those people need to be provided with correct nutrition, correct frequency, and correct quality of nutrition. Of course, you should provide probiotics, eubiotics, and different other drugs containing normal flora bacteria or vitamins and essential nutrients for restoration of normal flora. And in severe cases, if still you see that normal flora cannot be restored or toxigenicity is very huge, of course, you should add the same metronidazole as the main anti-anaerobic drug, or even bacitracin, unique antibiotic produced by Bacillus anthracis, which can inhibit other concomitant gram-positive bacteria and have only local activity in intestine. We have several methods of toxin identification. I will show you in next slides. It is photo of pseudomembranous colitis. In the left side, it is endoscopy, colonoscopy. You see those patches of yellow color covering the whole intestine. It is pseudomembranes caused by Clostridium difficile. In the right part of the slide, in the right picture, it is an autopsy material resected colon and the same yellow purulent containing non-mucous membranes. Of course, it will severely damage intestinal cell wall, lead to incorrect peristalsis, incorrect absorption of nutrients. So for identification, initially we had a ELISA test If you remember, it is a type of immunological assay when antigens of Clostridium difficile, in that case it is toxin A and toxin B, can be identified with pre-absorbed on that plastic well. Antibodies, complex antigen antibody can be detected with the help of specific enzyme, which is also fixed on anti-antibodies, and changing of color in that test system in blue color show you that sample is positive. So it is 96 plate well, 96 wells plate, and you can see that multiple tests were performed. Some blue, some remains transparent. So blue indicates positive test. But usually, in the case of ELISA, we can just identify only toxin. We cannot differentiate A or B. It's just showing that some toxin is present, and of course, it means that Clostridium difficile is the reason. But that test cannot show you presence of non-toxigenic strains. You'll identify only toxigenicity of Clostridium difficile. So, and test requires few hours, and nowadays mainly it is replaced by immunochromatography, and principle is kind of the same, but instead of plastic wells and some liquids, we have that specific plastic plastic strip with window for sample, for additional sample, and indicator window which is marked with the C and T letters. C is a control, T is a test. It is a unique membrane which can pass only known size of complexes, and actually the same similar principle. You need to take sample from the subject. You don't need any some pure culture. The test can be done near the bed of the subject and requires about some 15 to 30 minutes. You need to take a sample diluted with specific reagents provided with the test aid in the deliver sample window, and antigens of Clostridium difficile, if they are present, they will diffuse in that kind of paper membrane, and they will form a complex in obligate manner with the control sample because it is added with the reagents, and it's smaller in size. It can pass almost till the end of the strip and will be held by membrane and accumulate in the C zone. If it is present, that means that result is indicative and can be used. If antigen, some toxin or other antigens used for identification of C. difficile are present, they will form bigger complex that will be held by first membrane near the T letter, and you should have two strips. So if T is indicative, it is positive test. If C control is indicative, it means that result is correct. If you see that only test strip is appeared or nothing is appeared, test is invalid, it should not be used. You need to remake it again. So it is the real test system. Actually, we have a number of them. They can be as well for identification of Clostridium in general, combined test, which will show you only one result. It cannot differentiate. In current picture, we have even triple test. It is targeted against both toxins and unique enzyme, glutamate dehydrogenase, which is present in Clostridium difficile. And you see the three different strips, and lowest part when S is shown, it is a window for sample, and fecal matter is added, even some inclusions of pieces is visible in that drop of solution. And that water liquid is diffusing in membrane in paper strip, and in GDH window you have two strips, red and green. Appear that means that control window is working and test result is positive, means that Clostridium difficile is identified. But windows for toxin A and toxin B contain only green strip. It means that control is working, test is performed correctly, but toxins are not identified. So if bacterium be toxigenic, the same red strip should appear in one or both windows according to which toxin is produced. Test is simple, principle is the same. Antibodies against toxin are pre-added on that paper strip, and when you add sample, antigens will form a complex with antibody and they will diffuse in paper strip and will be held by membrane. So nowadays, identification of Clostridium difficile is very simple and fast, and those rapid tests are usually available in department with massive antimicrobial therapy. The only possible method of prevention of the disease is correct administration, and we call it rational administration of antibiotics. You need and you should monitor when you prescribe antibiotic and why you prescribe. If you prescribe an antibiotic, record in case history, what was the indication, which dose should be provided, how frequently, how many times per day, and how long patients should receive that antibiotic for further reevaluation. And usually we prescribe initially for 3 days, and if efficacy is good, we can prolong treatment for as many days as required. But please make a record in case histories that you reevaluated the result. Antibiotic is active, so I will continue some for 3 more days or 4 more days. And when antibiotic is not required, when reason is eliminated, stop antimicrobial therapy. And of course, combinations of antibiotics should be prescribed. Only is the most radical measure when single antibiotic cannot suppress your pathogen, your required pathogen. So vaccination, of course, is not possible because Clostridium difficile again is found in it is a part of normal flora of intestine, and it fine to have it. It's not a problem until it is controlled by normal flora bacteria. So you have one question about non-sporin anaerobes. It is a huge group, and actually, if in spore forming you have only Clostridium, and non-sporin anaerobes we have massive number of different microorganisms. They belong to any possible group. We have gram-positive copies, and usually we given some referates or additional questions and classes about that bacteria because they're not really so specific, and usually, actually we have now some enough information to provide, and properties are common. These usual anaerobe but with some unique appearance. So in gram-positive coccus, mainly you should know Streptococcus and Peptococcus. As you can see in the slides in microscope view, and is it obvious from the name, Streptococcus have arrangement in chains, short or long. It is a spherical shape, blue in color bacteria, and sorry, in the right picture should be Peptococcus, just Peptococcus. They are arranged in pairs or located singly in some groups, as well gram-positive coccus. So both microorganisms and actually almost any anaerobic bacteria usually is identified as a part of normal flora of GIT in different parts. So Streptococcus and Peptococcus can be identified in mouth and bowel. They can be recognized in flora of urogenital tract, and mainly diseases by anaerobic bacteria are rare and originate from subject itself after some surgery or after trauma, and location of bacteria nature location usually is the place of further infection. So both Peptococcus and Streptococcus can be identified as the etiological agents of urogenital infections, of appendicitis or internal abscesses, and very rarely they can spread via blood causing sepsis and some osteomyelitis and separative infections of other internal organs. The next group we need to discuss is gram-positive Rotae bacteria, and again, that bacteria is usually given for you in the form of referate. We have several of them. Mainly they belong to main digestive flora of bowel. It is Eubacterium, Bifidobacterium, Lactobacillus, and some other microbes which are naturally found in different parts of the bowel, and they perform very significant important digestive functions and secretion of some vitamins, and can perform other functions. In oral cavity, they can be associated with different periodontal diseases, with infections, operative infections of gums and soft tissues of oral floor. But again, usually It is observed after some surgical invasions in stomatology, in dentist practice, and after surgery on GIT organs. We have one unique bacterium which is, I think, most known in all anaerobes. It is Propionibacterium, and the main species is Propionibacterium acnes. It is causative agent of juvenile acne. It is type of infection of the skin with formation of small abscesses, or they can obstruct pores of skin glands and form those black dots on face. And usually that bacteria is widely used in commercials of different local lotions, creams to prevent appearance of those changes in the skin in young adults. As you can see on the slides, bacteria are looking like a little different. Bifidobacterium have very unique appearance. It's kind of like a snowflakes, and other bacteria can have different size, different shapes, and usually they're used in identification of dysbacteriosis and correction of some nutrition in different types of dietology. The most famous anaerobic bacteria in general is Bacteroides. It is the prevalent and the main digestive flora of large intestine in human. The main species represented in that genus is Bacteroides fragilis. It is most often associated with internal diseases in human. It is shown in left part of the slide, simple gram-negative rod, present usually in huge amount. They can be located singly or forming some small groups. That bacteria is usually associated with two types of infection. They can cause peritonitis after bowel trauma, after perforation due to some ulcers, or after interventions by different reasons in abdominal surgery. And second thing, they can be associated with urogenital infections due to anatomical close location of both systems, and can be associated with some internal abscesses, but again, usually that infection is associated with some previous medical intervention. We have Prevotell and Leptotrichia is a few bacteria known by, again, infections of gums and mouth. And Leptotrichia can cause a disease which resembles diphtheria by clinical presentations. It is called Vincent's angina, and it can have some unique medical significance. So, common properties of bacteria kind of common. They should be cultivated in absolutely strict anaerobic conditions. Any contact with oxygen will lead to immediate death. If you need to collect samples, it should be done very fast and without any contact with the air. And mainly, blood agar is used for identification and for cultivation of any genus and species of anaerobic bacteria, but you should keep it in specific incubator or anaerostat to prevent contact with oxygen. So the main problematic thing is how to take sample and not to have a contact with air or with environmental objects. So usually any blood sample in routine clinical practice should be studied for both aerobic and anaerobic bacteria. And as is shown in the left part of the slide, in the left picture of the slide you saw, you can see that row of bottles. It is different commercial test system for cultivation of anaerobic bacteria from blood. Needle is adjusted directly through cap of the barrel, plastic part is removed, and rubber part is in the barrel. You need to pass needle through it, and it contains negative pressure, have negative pressure and kind of vacuum. When you will perform injection in vein and adjust that barrel, it will suck the blood automatically and without any contact with the air. So later that bottle should be put in an automated device, and it is already suitable for cultivation culture medium, and bacteria can easily grow directly here. You don't need to perform any re-inoculation in another culture medium. So in the center of the slide, it is a swab. Again, commercially available swabs with unique plastic tube containing gel in consistent culture medium. After taking sample with that cotton swab, you need to inject it through plastic part in the gel, and gel will collapse and perform anaerobic conditions. It is a transport swab to deliver sample in laboratory. And in the right part, it is a culture medium, which is known as a Robertson cooked meat or Kittarozzi medium. They are very similar by principle and by general appearance. It is a broth, meat broth with the samples, with the pieces of some meat or other biological tissues. So as well, test tube can be easily closed, completely closed. It is a metal cap with the rubber inside, and some oxygen which will present in the upper part of the test tube will be absorbed by biological tissues, and anaerobic conditions will be provided in the test tube. It can be used as a transport culture medium, or in some laboratories that culture medium is used for cultivation of bacteria. It will grow as a turbid film in the near the bottom or on the surface of those tissues. So if you discuss any anaerobic infection and therapy of it, the main idea that non-sporing anaerobes can be easily killed by oxygen, and very often main type of therapy is oxygenic therapy. People can be put in hyperbaric oxygenation chambers, or just some catheter with oxygen should be put into the wound. Wound should be opened and provided with oxygen access. And antibiotic, usually it is one metronidazole, which covers the whole spectrum of anaerobic bacteria, is widely used in any hospital in routine practice. So that's all for obligate anaerobic bacteria.